If you guys want to go ahead and grab your folders out too, if you haven't done that yet. Grab your folders out. So, for the most part, we did a pretty good job on the test questions regarding the structure, the diagrams of how DNA is built. But 8, those dashed lines between our bases there, those are hydrogen bonds. This circling the 3 parts of a nucleotide. 10 is pointing to A, which is an example of a base, nitrogenous base. It's going to be more specific. Yep. 11. These are the ones that are, I think, the easiest to mix up. But 11 is specifically a deoxyribose, and you can tell because that's the part that's connected to the base, right? So the sugar is connected to both the base and to number 12, which is the phosphate, right? And it's these, like we mentioned yesterday, it's the code, the base sequence on one of those strands that's eventually going to be able to code for our proteins, right? So we spent some time yesterday kind of reviewing some information about protein structure and why proteins are important. Did you know there's a protein that makes light? I mean, so proteins do all kinds of different functions in living things. It's pretty neat. And it comes from just this really simple 4-letter code, right? So we're going to dig a little bit more further into the protein process, kind of an overview today. And we're also going to talk about RNA molecules a little bit. So what are some similarities, importance to that? Because RNA plays a huge role in that transition from taking the code in DNA and making it into protein. So if you open your folder to the next page for notes. By the way, I did notice there's some people that didn't get the Canvas quiz for the protein scavenger hunt yesterday done. So again, make sure you don't get too far behind on those assignments. They're half your grade. Chapter 8, Part 2. That's where we're going to start page number 3. And this first slide here is not going to be on the fill-in notes, just something to listen to. And so, the whole part of this chapter is to figure out, again, how does that 4-letter DNA code end up eventually forming proteins? And those proteins are then going to build all of our cells, and our cells, of course, are going to build us, right? So the DNA codes for a protein, our protein does all the work and builds our structures. And so essentially, when you look at each other, when you look at the person next to you, all the differences that you see are because you make slightly different proteins, and you make slightly different proteins because you have slightly different DNA. 99.9% of your DNA is exactly the same as the person next to you. Not that 0.1% difference it's probably even less than that, is what causes us to have different traits. So the central dogma, this is what is shown on your notes here. The central dogma is in biology is the pathway of genetic information, the way that DNA is used to make proteins. And so you got to be a little bit careful in correlating this to your diagram here, but On the left-hand side, in that circular arrow, we have DNA, right? DNA is the start of this pathway. It's where all the genetic information is stored. And last section, we talked about how we can make DNA through that process of replication, right? So we can replicate our DNA, and we can then pass that on to other cells. From DNA to RNA, that process is called transcription, so we'll probably start talking about that more tomorrow. And that RNA then is used to do the process of translation, which is going to put together our protein. So there's some big important steps between DNA and protein that involves this molecule called RNA, ribonucleic acid. And so to help us kind of think about this pathway, we're going to do an analogy here on the next slide. DNA gets all the glory, but the proteins do all the work. So, in our analogy here, our nucleus is like a safe, and inside of that safe, we are storing our DNA, which is our blueprint. Right? So our DNA is like a set of blueprints because it holds the instructions for building proteins, right? Or therefore for building our cells, and therefore for building our bodies, right? So the blueprint for us is coded in that DNA. And the nucleus is like a safe, because we have that for protection, right? We're protecting our DNA from getting hurt or damaged. Still can get damaged, but the nucleus helps to prevent that. And so why does DNA have to stay in the nucleus? So go ahead and talk about that in your group really quickly. What do we want to the point of a safe is to keep the DNA from getting out and getting damaged, right? So we want to keep the DNA safe. We don't want to damage our blueprints, right? Because if we mess up those copies, then we don't have the right instructions anymore. Used to be way back in the day before Google was a thing, you'd have to go to the reference part of the library, and that's where they would store all the encyclopedias and the important books, and you're not allowed to take those books out of the library, so you had to copy information from it. So now that analogy doesn't really make sense to a lot of people, but that's kind of what it's like. So we take that DNA blueprint, and it has to stay in the nucleus. So in order to use it, what do you think we have to do with that information? We have to copy it. Okay? So that copy is there's multiple different forms of RNA, but when we want to make a protein, the message is coded in that mRNA. So the mRNA is just like a copy of the blueprint. So we can make as many copies as we want, as the cell needs. If one gets damaged, it's not a big deal, because we can just make another copy. But as long as our original is safe, then we don't have to worry about anything else happening to it. So this step between DNA and protein, making this mRNA, that's making copies of the instructions, so that we don't have to worry about messing up the original. So these are disposable copies. We don't have to worry about them getting damaged. So those molecules are in fact going to leave the nucleus and go out to the cytoplasm. That process of making the copies is again called transcription. Just transcribe something just means to make a written copy of it. That mRNA is going to go to a ribosome. Anybody remember anything about ribosomes? One tag off. Do you remember something, Ethan? Yeah, that's where the proteins actually get made. So the copy of the blueprint is going to go to the ribosome, and you can kind of think of the ribosome as our protein factory. Okay, so at the ribosome, we're going to use the copy of those blueprints. We're going to use the instructions to build our protein. And notice when I animate this protein on here, it's just a series of these little circles with an A on them. What do you think that A stands for? No. What are proteins made up of? Amino acids. Yeah, so when we make the protein in the ribosome, really all we're doing is putting a bunch of amino acids in the chain. We're putting all these amino acids together. And then that's going to fold up in a particular shape, right? And that shape is then going to go on to have a particular function. So we'll get more to that later, but that's our representation of our protein, all our amino acids that are getting put together. That process, so there's the final structure of a protein, right? That process is called translation, because at this point, we're basically going from a different language. We're going from nucleic acid language, RNA, to protein language, amino acid. So, the next couple slides, we're going to talk about some important differences and similarities between our two nucleic acids here. So DNA, our blueprint that we don't want anything to happen to, and mRNA, our copy of the blueprint that we can use and not have to worry about damaging. The proteins, sorry, I got a little bit ahead of myself. Those proteins are then going to determine all of our traits, our eye color, our hair color, our height, our blood pressure, how well we carry oxygen, how well you digest different materials. Like everything about you is determined by these proteins. So, RNA versus DNA. First really big difference. We know DNA is double-stranded in a double helix. RNA, on the other hand, is just a single strand. So just one string of nucleotides. So DNA double-stranded, RNA single-stranded. They're both just chains of nucleotides, though, right? So they're both nucleic acids. They're both have polymers of nucleotides. You'll notice here it says helix of sugar phosphates. So that backbone there is all sugar and phosphates. And then here's our bases. RNA is the same thing, sugar and phosphates and bases. Okay, it's just one half of the helix, one strand versus two strands. Because they're both nucleic acids, their building block, their monomer is very similar. They're both made up of nucleotides. Okay. So take a couple seconds there, see if you notice with your group what's the main difference between DNA nucleotides and RNA nucleotides. Yeah, good. It's a different sugar, right? The name DNA stands for deoxyribonucleic acid. RNA is ribo nucleic acid, because there's a slight difference in the sugar, right? This is deoxyribose in DNA, ribose in RNA. So there's an extra oxygen here in the ribose that's not there in the DNA that makes it just structured just a little bit different. So both made up the nucleotides, sugar, phosphate, base, but DNA has deoxyribose as the sugar, and RNA has ribose. That's where the difference in the name comes in. Right. Here's some bases with your group. What's similar, what's different? Yeah. A lot of the bases are exactly the same, right? Both have A, both have adenine, both have guanine, G, both have cytosine, C. But here's the difference. In DNA, the basis here is thymine, and RNA is a very similar structure, just slightly different. This is uracil. So in RNA, the thymine base is replaced with uracil. What do you think uracil probably paired with? Adenine. So it literally is replacing any thymine. What we would have as thymine in DNA is not going to be uracil in RNA. Okay, the base pairing rules are going to work exactly the same. So in our base pairing rules here, we're used to seeing A pair with T. RNA instead of T has U. So for pairing from DNA to RNA, it's going to go A pairing with U instead of A pairing with U. But all the other matches are going to be the same. If I'm matching RNA with DNA, if I have a T in DNA, I'm going to pair it with an A of RNA. If I have a C in DNA, I'm going to pair it with a T in RNA. And lastly, if I have a G in DNA, I'm going to pair it with a T. So the base pairing rules are almost exactly the same, except where I would put a T. If it's RNA, it's going to be a U. Make sure you guys are filling something in about that on your. Look back out here. So if you haven't looked underneath that chart there, there's this problem. So go ahead and take a couple seconds to think about that and fill that in and talk about it in your group. Last test, we talked about replication. So if this is my DNA strand and I'm going along and adding DNA nucleotides, right, to make my copy, I had an A, DNA polymerase would come along and add a T. And for T, we would add an A. And for G, we would add C. And for C, we would add C. And for A. And for T. And for C. And for G. C. Perfect. Because we know those base pairing rules, right? If instead of replicating, and instead of adding DNA nucleotides and making more DNA, we were going through the transcription process, which again we'll talk in more detail about tomorrow. Instead of adding DNA nucleotides, we're adding RNA nucleotides. So instead of a T here, I would have a U. And everything else would be the same though, right? T is still going to pair with A, and G is going to still pair with C, and C is still going to pair with E. But again, if I hit the next A, it's going to be a U. Okay. A, G, C. And so almost identical, except I don't have thymine and RNA, I have uracil. So that's the notes for today. I would like to do test corrections, so I have your test graded. So I'm going to keep your folder out. The other thing when you're done with your test corrections is there is another Canvas quiz over the material we just covered. Okay, again, just to make sure and check that you got that information and you know it. So after you do your test corrections, if you want to pull out that Chromebook and take that Canvas quiz, I would advise it. Okay. So just like last time we did test corrections, I'll call your name. You can come up and get your test, and don't forget to get the actual test itself too. So your answer sheet and the test, so you can actually fix the questions. And you'll have this strip that'll tell you which ones you need to fix. So I got Jadlyn. Come on up. Beck. Hopefully I'll give you one strip this time. Mia. Kaden. Ryan. You don't have to correct your essay questions, but I did leave comments on some of them, so. Emily Baldwin. Allie. Starlet. Parker. Here. Oh yeah, because you only pointing for that. Okay, yeah, sorry, don't worry about that. Thank you. Yeah, do whatever you want with that. Helix. Connor. Kevin. Emily Stuckey. Madison. Murphy, sorry. Maddie. Naomi. Ethan. Madison. And Reese.